Hi everybody, Assalamualaikum warahmatullahi wabarakatuh. Welcome to Food Technology Study Program, Department of Food Science and Technology, Faculty of Agricultural Engineering and Technology, IPB University. My name is Rati Dewanti Haryadi. I am a professor in food microbiology at IPB University. Welcome back to ITP 322 Food Safety and Sanitation course. Today is lecture 9 and the topic will cover personal hygiene, air sanitation, and pest control. Today's subject will cover three areas: personal hygiene that will include potential sources of contamination from human, effective hand washing program, and prevention of cross-contamination. And the second subtopic is air quality and food safety, in which we will be discussing bioaerosol, direct and indirect contamination, and air sanitation. Lastly, we are going to discuss pest control, which will cover permanent control action, supplementary control application, and guidance of pesticide application. Upon completion of this lecture, you are expected to be able to explain the role of workers as the source of contamination in food processing, explain effective hand washing, describe cross-contamination, describe bioaerosol, relate bioaerosol and food safety, and compare different air sanitation methods, as well as describe pest control program. Let's begin our discussion with workers in food industry. While they are important part of food industry, they also indeed are potential sources of contamination. Skin, for example, has been known to contain residential bacteria. Usually they are not pathogen, but they live and grow on the skin, such as Micrococcus luteus and Staphylococcus epidermidis, and transient bacteria that occasionally occupy and colonize skin, such as Staphylococcus aureus. We also can find bacteria in fingers, fingernails, hairs, eyes, mouth, nose and nasopharynx, as well as respiratory tract usually also contain viruses, and intestinal tract usually contain Escherichia coli and other enteric pathogens. Jewelry worn by workers may also carry some bacterial pathogens. In food industry, the main objective of personal hygiene program are to prevent product cross-contamination and to prevent workers from getting infection. The personal hygiene program could include hand washing program, rules on jewelry, control of hair and beard, use of protective clothing, apron, hair cover, shoes, etc., and control of behavior such as eating, drinking, smoking, etc., as well as other regulation concerning the cosmetic or jewelry. The following are several activities concerning control of diseases and cleanliness in personal hygiene program in food industry. For example, taking a bath daily before working. Hair should be washed at least once every week. Nails should be kept clean and cut. No jewelry around production area. Communicable diseases such as TB and hepatitis viruses should be reported. An employee with open wounds or infected skin should not be allowed to handle food product directly. If there is a wound on hands, cover it with bandage and use disposable gloves. To prevent cross-contamination to the product as well as to maintain their own health, workers has to be trained how to wash the hand properly. Washing hand properly shall include removing all jewelries, wetting hands with water, adding antiseptic soap and rubbing it for a minimum of 20 seconds, rinsing it, and dry it with paper towel or hand dryer. Workers in food industry also have to know when to wash their hands. After touching part of their bodies, after using toilet, after coughing, sneezing, using handkerchief or disposable tissues, smoking, eating, or drinking, after handling of dirty equipment, and during food preparation as frequently as required to remove contaminants and prevent contamination that could cause cross-contamination. Therefore, food industries have to provide hand washing facilities that are equipped with antiseptic soaps containing germicidal compounds, suitable sanitizers, which are frequently monitored, cleaned, and changed, and placed on reachable locations. To prevent cross-contaminations, workers should also be equipped with protective clothes such as uniforms, head covers, masker and beard covers, and clean gloves. The following are symptoms and conditions of concerns among the food workers. People with diarrhea, sore throat and fever, skin open wound, vomiting, pus wound, and yellow eyes or skin, as well as dark urine, shall not be allowed to handle food directly. Employees may also be carriers, that is, people who carry enteric pathogenic bacteria such as Salmonella typhi, Shigella, etc., without showing any symptoms. These carriers can be differentiated into three types. The first one is convalescence carriers. These are people who just recover from infectious diseases and continue to harbor the organisms for variable length of time, usually less than 10 weeks. The second type is chronic carriers. This group containing people that harbor the infectious organisms indefinitely without showing any symptoms of diseases. Contact carriers are people who acquire and harbor a pathogen through contact with an infected person, but they do not acquire the disease or show symptoms. In conclusion, personal hygiene programs in food industries aim to prevent cross-contamination, that is, transmission of biological or chemical contaminants on food products from raw materials, from employees' hands, or food handling environment. Some of the preventions include personal practices to prevent cross-contamination, separation of raw materials from ready-to-eat foods, and plant designs to prevent cross-contamination, which are usually achieved by zoning or separation of areas in food industries based on the risk. We have discussed zoning when we discussed sanitary design for building and facility in Lecture 8. And we also know that other factors that also contribute to the quality and safety of food products are room temperature and humidity, as well as room airflow and air quality. So what is the relationship between air quality and food safety? Outdoor air has 200 to 1500 bacteria per cubic meter. A 10,000 meter cubic per hour air conditioning system could take in about 2 to 15 million bacteria per hour. Therefore, food industries use air handling unit, air filters, HVAC filters to reduce the risk of airborne microbiological contamination by preventing microorganisms from entering food processing facilities. However, the role of airborne contaminations to the quality and safety of processed food is controversial. Some think that airborne contamination of food is of public health significance, but some don't. So let's talk about bioaerosol. Bioaerosols are solid or liquid microscopic particles suspended in the air that carry microorganisms. A contaminated aerosol, whether liquid or dry, is comprised of injured bacterial cells or spores. Where are these bioaerosols came from? It can come from food, wheeled equipment, vehicle traffic through standing water, application of high-pressure washers to contaminated surfaces, and use of compressed air line without appropriate use of filters. Bioaerosols are suspended in the air for various times. and the settling rate is depending on the square of the particle diameter. The vegetative cells in the air will be diluted and usually are injured. They move from high to low pressure on air current in the plant. Therefore, it's important to have appropriately filtered positive pressure air in the rooms that require higher hygienic controls. Recovering pathogens or microorganisms in bioaerosols from the air is difficult. It requires large volumes of air imping for an hour in enrichment medium. It is prudent, though, to monitor airborne microbial population for hygienic indicators and take appropriate corrective actions when exceeding the acceptable level at the factory. Contamination of airborne pathogen to food can be differentiated into direct and indirect contamination. Direct contamination of food from air may or may not be significant depending on the type of the food products. When bioaerosols contaminate a product with intrinsic or extrinsic inhibitory properties, such as low water activity, low pH, detectable airborne contamination will not occur. However, when bioaerosols contaminate a product conducive to microbial growth, measurable airborne contamination will occur given enough time at an appropriate temperature. For example, contamination of milk by airborne psychrotropic Pseudomonas, or fruit juices by Alexiklus bacillus, or vacuum-packed raw meat by Clostridium aestheticum. Indirect contamination occurs when trace numbers of undesirable microorganisms may be present in the air and contaminate areas in the plant conducive to growth, for example, in the standing water, sandwich structures of equipment that entrap wet residues, and recovery of injured cells may occur. High population of microorganisms may then develop in the above area, initially inoculated from air, drip, drop, slough, or otherwise be transferred to the product. Then direct measurable contamination become more likely, even in food that is not conducive to microbial growth. In general, air sanitation is the system of removing the impurities present in air inside buildings to protect people from infections. Sanitation of air is essential in food processing and packaging industries and rooms where sterile materials or products are stored. Factors that determine how conducive a particular building is to spreading diseases include the following: the range of temperature and humidity control, the amount and distribution of outdoor air, the efficiency of the filters, the cleanliness of the facility, the number and the types of surfaces throughout the buildings, as well as the hygiene of the occupants. Here are some devices for air sanitation. The first one is ventilation. A ventilator is an opening or device that allows fresh air to enter to an enclosed space. A great reduction in the airborne microorganism can be carried out by laminar airflow system. A horizontal flow of air at 40 feet a minute in a room in which spores of Bacillus subtilis had been liberated led to a decrease in their number by 100 to 1,000 times greater than that in a room where turbulent ventilation were employed. The second device for air sanitation is safety cabinet. Safety cabinets are isolation chambers providing a safe environment from pathogenic microorganisms. Negative pressure air protect from bioaerosol within. There are three classes. Class 1 exhaust protection cabinet is aimed to protect the operator by maintaining a rapidly moving stream of air, entrance infectious particles, and deposit them on a high-efficiency filter. Class 2 laminar flow cabinet is an open-fronted designed to protect materials from extraneous contamination. It is designed for moderate pathogens and do not protect the operator. However, it can be modified to do so. Class 3 cabinet is for very dangerous materials, completely enclosed. Materials are handled through gas-tight glove ports, and air enters through a non-return valve and is extracted through a high-efficiency filter. The other device for air sanitation is disinfectant sprays and vapors. In hospital, carbolic spray, sulfurous acid, and formalin can be used for terminal disinfection. However, they are highly irritating to respiratory passages. Gram-negative bacilli are more susceptible to UV light and disinfectant vapors than gram-positive bacteria. Microorganisms are harder to kill when dried onto dust particles than when they are in the form of droplet nuclei. Sodium hydrochloride is effective to destroy E. coli in the atmosphere. Ozone and 2.5% hydrogen peroxide is also effective for killing Salmonella typhi, while resorcinol, propylene glycols, triethylene glycol, and ozone are not considered effective. The following are technologies that have been developed or are being developed for airborne pathogen control, and we are going to discuss some of them in the following slides. One of the current air disinfection methods is isolation systems. This system can classify the rooms into three basic categories: negative pressure isolation room, positive pressure isolation room, and multi-level biohazard laboratories. The second method is filtration of microorganisms, for example, by using high-efficiency particulate air or HEPA filters. These HEPA filters can be box or pleated and bag type, consisting of fine fiber, synthetic fibrous materials, and made of various materials. They can effectively remove dust and particulate above 0.3 micron. Highly effective for bacteria, although it's fairly effective only for viruses. It has been used in hospital isolation room, operating theaters, level 3 and 4 containment facilities, and industrial clean rooms. Here are the principles of filtration of microorganisms using HEPA filter. The filter does not restrict the passage of particulate. Instead, it alters the airflow streamlines, thus airflow will slip around the fiber. Any higher density bioaerosol or particles will not change direction so rapidly, and as a result of their inertia, they will tend to impact the fiber. Once they are attached to fiber, the particulate will not re-enter the airstream. Another method for disinfection is the use of UVGI or ultraviolet germicidal irradiation. Light at or near 253.7 nanometer can break molecular bond, cellular, or genetic damages in microorganisms. Viruses are more susceptible than bacteria, and here are some factors affecting the effectiveness of the ultraviolets: exposure time, room air mixing, presence of moisture or particulates that provide protection for microorganisms. Dust settling on the light bulbs can reduce exposure, thus maintenance is necessary. Now here is some technology under development for air sanitation. Photocatalytic oxidation using titanium dioxide irradiated with photon to produce hydroxy radical and superoxide ions, which are highly reactive to oxidize volatile organic compound or VOCs adsorbed on the catalyst surfaces and kill and decompose adsorbed bioaerosols. PC reactors can be integrated to HVAC system, and these attributes contribute to the potential of PCO technology to be an effective process for removing and destroying low level of microorganisms in indoor air, including bacteria, viruses, and fungi. Another technology under development is air ozonation. Ozone is injected into the airstream and mixed in the turbulator that allows ozonization of all organic compounds, including viral nucleic acids and bacteria. Due to the corrosiveness of the ozone, though, an efficient reclamation system must be developed. Reclaimed ozone could be recycled to the injector or neutralized and used to regenerate electricity to feed back to the regenerator. Ozonization has proven extremely effective in water systems, but as yet no air-side system has been developed and proven to be effective. Lastly is carbon adsorption. This system is used primarily for removing gases and vapor and effective against VOCs, but not for airborne dust or microorganisms. The system consists of materials, for example, activated charcoal with enormous amount of surface area, therefore allow gas molecules to adhere to the surface. It is unlikely to have a significant effect on airborne microbes, but can be effective at removing VOCs generated by fungi and bacteria. The use of carbon adsorption for the control of airborne viruses, which is similar to VOCs, is a potential application. A tenfold increase in pore size might be sufficient to adsorb viruses. Our last subtopic is pest control. Pests in food industry can carry foodborne pathogens. Flies and cockroaches has been proven to transfer pathogens such as Salmonella, Staphylococcus, Clostridium perfringens, etc. Rodents are the sources of pathogens such as Salmonella and parasites, and birds are the host of various pathogens including Salmonella and Listeria. Pest control program in food industry can be done in a three-step pest control program, which consisting of eliminating all shedders and attractants, removing pests out of food plant, and eradicate pests already entered the food plant. You can also have a complete pest removal program, which require monitoring the presence and absence of pests and monitor all of the related conditions such as plant and premises, structure and layout, plant, machineries, equipment and utensil, housekeeping, waste removal, and application of pesticides. Rodent and insect control in food industry is built with basic sanitation and then a permanent control and if necessarily a supplementary control. Basic sanitation includes all activity that assure the cleanness of the food facilities, such as several activities you can see in this slide. Permanent control for rodents include rodent-proof facilities, removal of food residues, and elimination of rodent infestation sites, while for insect include proper building construction and maintenance, good housekeeping and warehouse practices, as well as control of environmental factors. The supplementary control action for rodent and insect and food industry is the use of pesticides chemicals such as rodenticides and insecticides. In Indonesia, the production, distribution, and application are controlled by the Pesticide Committee of the Ministry of Agriculture in Indonesia. Many pesticides are limited for specific usage and application method. Some has to be applied only by a certified applicator. Here are some of the guidance for pesticide application. Pesticide agents shall be applied as such not to contaminate the edible product through open windows, ventilation system, etc. Before application with fumigants, all edible products and packaging materials should be removed from the area. After fumigation, the treated equipment and area should be well aerated to eliminate all gases before the employees enter the area. Food contact surfaces should be rinsed with drinking water before edible products are placed back to the area. Don't forget to work on your group presentation assignment for next week for Lecture 10. Thank you for participating in this class. We'll see you again next time. Assalamualaikum warahmatullahi wabarakatuh.